So this is one example of competitive inhibition, whatever we have discussed. So one is methotrexate, second is aminopterin, which are generally used as anti-cancerous drugs. So I'm reading whatever is written in this book. So in competitive inhibition, Vmax will stays the same and Km increases. You need to remember this each time and always. So please underline this: Km will increase, but Vmax will not change in competitive inhibition. But the inhibitor does not affect the turnover number of the enzyme, because the same efficiency can be achieved with the same enzyme which is undergoing inhibition. If you just increase the substrate concentration, and everything will be normal. Clinical treatment of methanol poisoning, which we have studied yesterday with the help of ethanol. So if somebody have consumed methanol, which is also called sasti sharab, so you can treat that particular patient by providing him or her pure ethanol. So that will be the pure substrate of the alcohol dehydrogenase that will replace all the methanol from the enzyme, and therefore the harmful product, that is formaldehyde, which is generally harmful for soft tissues of our body, especially your eyes. So it has been seen that the people who consume it is by chance, obviously, nobody is going to consume the methanol intentionally. So this is harmful. Formaldehyde will be formed, and this is the product of alcohol dehydrogenase when it will bind with methanol. And this will harm your eyes, and those particular patients are undergoing some blindness. Okay. A high dose of ethanol is generally used to alleviate the effects of methanol, because ethanol competitively bind to the active site of the alcohol dehydrogenase enzyme, and therefore the ethanol will be converted into acetaldehyde instead of formaldehyde. And acetaldehyde is not that much toxic to our body, so therefore it will do no harm to. It will harm, but that is not so much severe as, for example, blindness. Similarly, methotrexate, this is the second example we have recently discussed. So methotrexate is an anti-cancerous drug and potent competitive inhibitor of the enzyme dihydrofolate reductase, which is playing a role in biosynthesis of nucleotides, purine and pyrimidines. Methotrexate acts as a structural analog of dihydrofolate. So you should also remember that the normal substrate of dihydrofolate reductase enzyme is dihydrofolate, which will be converted into tetrahydrofolate in the presence of NADPH. Okay. So after this, since it is very visible from both the graphs that the Km is increasing and the Vmax will remain the same, because they are intersecting at the same point. So if this type of curve you face somewhere, these are competitive inhibitors. Okay. Then second one is non-competitive inhibition. And I told you that non-competitive inhibition may be of two types: pure non-competitive inhibition, in which you can assume that this alpha is equal to alpha dash, means this alpha. What is alpha? 1 plus I upon ki, and alpha dash is 1 plus I, that is the inhibitor concentration, upon ki dash. What is ki and ki dash? These are the rate constant of these reactions, because in non-competitive inhibition, either the inhibitor will bind to the free enzyme, or the inhibitor can also bind with the enzyme substrate complex. Also, this enzyme inhibitor complex can bind with the substrate to make ESI complex. So all the possibilities in mixed, therefore it is called non-competitive mixed kind of inhibition. So since there are two types, so first of all we will discuss pure non-competitive inhibition. It will occur when there is means both the constants, rate constants, are equal. Either enzyme, that is free enzyme actually, will bind with the inhibitor, or your enzyme substrate complex will bind with the inhibitor. In both the cases, the rate constants are equal. So in such case, these cases are called pure non-competitive inhibition. Now you need to remember, if this is pure non-competitive inhibition, so only Vmax will decrease. So whatever we have studied in competitive, here because there in the competitive inhibition, Vmax is not changing. Here, Vmax will decrease. There, Vmax is not changing. Here, Km, and in the case of competitive inhibition, Km was increasing. Here, Km will not change at all. It will stay stay constant. Okay, so the equation will be modified as, because we know the equation is normal, V is equal to V max into substrate concentration Km plus substrate concentration. So what can happen, because we know alpha and alpha dash are equal, so you can multiply the Km value and the substrate concentration with the same number or the same unit, that is called alpha. So this is apparent Km. So when you divide this V max by this, so ultimately this V max will decrease. If you rearrange the same equation in the form of double reciprocal LB equation, so what can happen, because Vmax is decreasing, so here also you need to multiply the Km value with alpha, also because this Vmax is decreasing, therefore you need to divide. Okay, so you will divide this Vmax with alpha, or in the other form, alpha will be divided by Vmax, because this is reciprocal equation. So normally, what is the LB equation? 1 upon V Km upon V max, 1 upon substrate concentration into plus 1 upon V max. Here, because V max is decreasing, therefore you will put alpha value here. Also, you will put 1 alpha with the Km also. But overall, in the pure non-competitive inhibition, there is hardly any change in the Km value. As we will see from the plot also. So plot is more clearly showing that Km is not changing. It will remain the constant. So suppose this is the Km value. In both the cases, the Km will remain the same, whereas the Vmax is decreasing. So it was the in the absence of inhibitor, minus inhibitor means there is no inhibitor. In the presence of inhibitor plus inhibitor, so this is a dashed line, so it will decrease from here to here. But the decrease in such a way that in the first case, this was a Vmax, so here was the half Vmax. If you plot this, so here you will get the Km value. In the second case, if this is Vmax, so if you try to find the second Vmax, it will come here. And if you draw these lines, they will come at the same point. Therefore, you cannot say that the Km is changing. There is hardly any change in the Km value. But the Vmax is overall decreasing. The same results can be more effectively seen from the line equation. So this is the benefit of line plots. So this double reciprocal LB plot, here there is no change in the Km value, therefore they both will intersect at the same point over x-axis, because x-axis is having 1 upon substrate concentration. This is the reciprocal value of substrate concentration. So the Km is also signifying the substrate concentration, so will come over x-axis, but it is a negative sign, so it will come this side. But you need to remember this, there is no change whether there is inhibitor or there is no inhibitor, both will intersect here, so the Km will remain the same. But because the Vmax is decreasing, so because we are taking the reciprocal value of rate 1 upon V, so if the Vmax will decrease, the curve will, or this line will, slope will move up. So the if you increase the concentration of inhibitor, so it will move further up. Okay, so that's how it will change. So Vmax will decrease. We are moving up, but because this is the reciprocal value, therefore Vmax is decreasing. Okay, now you can think of the mixed type or the mixed non-competitive inhibition, which is the second type of non-competitive inhibition. And because all the three are types of reversible inhibition, so this is also a kind of reversible inhibition. So in reversible, we are having three types: competitive, non-competitive, and uncompetitive. And the non-competitive is of further two types. One is pure non-competitive, one is mixed non-competitive. So that all we have discussed in yesterday's class, so no need to go into the details. So second type is mixed non-competitive inhibition, because I told you that it is also possible that one more reaction can happen here. So that this enzyme inhibitor complex can be converted into enzyme substrate inhibitor ternary complex. So this is the case of a mixed inhibition, and because this is mixed, therefore this alpha value will not be equal to alpha dash, or simply the rate constant ki will not be equal to ki dash. So one more diagram you can find from the Leninger. So it is better represented here. Uh, Actually, in case of mixed non-competitive inhibition, so these are the possible reactions which can happen. So this enzyme inhibitor complex, when bind with the substrate, can also be converted into enzyme substrate inhibitor complex, that is the ternary complex. And this Ki will not be equal to Ki dash. Both the rate constants are not equal. Whereas in the pure mixed inhibition, they were equal and they are not influencing the substrate binding. But as we have discussed yesterday, both the binding sites of this substrate and inhibitor are very close proximity. Therefore, they will affect each other. So if the inhibitor will bind, it will not allow the substrate to bind at all. Now see, okay, here. So first of all, because here you will see there is a change in Km value, and this change is not always increasing. This can be decreasing also. Overall, you need to say this Km value will also change. And you need to remember that the Vmax will always decrease in case of mixed non-competitive inhibition. So these two points are important to remember here. So how we will explain with the help of these two plots. So both are LP plots, double reciprocal plots. So they have plotted 1 upon V versus 1 upon S in both the cases. Now assume this Ki, what is Ki? So Ki is actually pure, sorry. So here, what is Ki? Ki is the rate constant when free enzyme is binding with the inhibitor. Whereas Ki dash is the rate constant when the substrate complex or enzyme substrate complex is binding with the inhibitor. So both are not equal. So see here again. If this Ki is when inhibitor is binding with the free enzyme. So this Ki is less than Ki dash. So you will find this cross here. So this cross here means your Ki value means the free enzyme which is binding with your inhibitor is having less reaction velocity in comparison to Ki dash, or the enzyme substrate complex is able to bind with more inhibitor. So in this first case, you will get a cross here. So this X shape will be received. And you will see here, if this is the Km in first case, in the second case, this Km value is generally increasing. So this is an increase. So I can plot it like this. And because this Vmax is decreasing, so it will move up. So in the first case, when this Ki is less than Ki dash, Km will increase and Vmax will decrease. Therefore, it is called mixed non-competitive inhibition, because one is increasing, one is decreasing. In the second case, if this Ki dash means this enzyme substrate complex when bind with the inhibitor is having high reaction rate in comparison to the free enzyme is able to bind with the inhibitor Ki. So in this case, this value will shift here. So here you will, so this will also give a V-shaped plot. Actually, this type of plot we were actually getting in the in that case pure non-competitive inhibition. But here, if you look closely, so actually these are not the true data values. So here actually on the x-axis you will get these Km values here and here. And if you see, actually this Km value is moving this side. So you can say that because this is moving more towards more negative, from the 0 to more negative left hand side, therefore the Km will decrease in this case, whereas the Vmax will also decrease. So both are decreasing. Km is also decreasing, Vmax is also decreasing if this Ki dash is less than Ki. So both can happen, both are the cases of mixed non-competitive inhibition. Either Km will increase or Km will decrease, but Vmax will always decrease in case of non-competitive inhibition. Hope this is understandable. If not, please ask me, raise a query. You can write in the chat box. I will explain it again to you if it is not understandable. Actually, this is very easy to understand. In the first case, in competitive inhibition, the Km was increasing. There is no change in the Vmax. But the second one is non-competitive. Non-competitive case, this Vmax will always decrease. In the pure case, there is no change in the Km value, but in the mixed type, there may be an increase in the Km value, there may be a decrease in the Km value. So the Km value will change and Vmax will also change. Therefore, it is called mixed non-competitive inhibition. Now we will come to uncompetitive inhibition. So in uncompetitive inhibition, you need to remember here the possibility of Ki dash only, because your inhibitor is able to bind with the enzyme substrate complex only to make a ternary complex. The inhibitor cannot bind with the enzyme here. Free enzyme cannot bind to your because binding of the substrate will lead to some conformational change in the enzyme so that it will be able to bind to the inhibitor also. Okay, so in uncompetitive inhibition, an inhibitor bind at the site which is distinct from the substrate. It is different from the substrate, obviously. And if you want to see here, it is also given here also. So there are two sites, one for the substrate, one for the inhibitor, and they are they are having some gap. Whereas in mixed inhibition, there is no gap between these sites, therefore they will influence each other. Okay, so here they are having some gap, but this will not bind to inhibitor until and unless the substrate will bind to it, and it will lead to some conformational change in the overall enzyme so that this inhibitor can bind more effectively with this particular enzyme. Fine, then we will move back to Pathfinder. However, an uncompetitive inhibitor will bind only to the enzyme substrate complex. We have discussed this. On the other hand, non-competitive inhibitor bind to either the free enzyme or the complex. This also we have discussed. So here, what is the major thing you need to note? Here, the inhibition, the Vmax will change and Km will also change, and both will decrease as in the case of mixed non-competitive inhibition. Okay, second one. So the apparent Vmax and the Km both will decrease. So this will decrease, this will decrease. We will see with the help of plot also. So first of all, you need to remember both are decreasing in uncompetitive inhibition. Okay. Then this is happening. So because there is a constant rate, constant Ki dash. So substrate, enzyme substrate complex concentration plus inhibitor concentration divided by the product ESI, which is a ternary complex. Now, in the presence of uncompetitive inhibitor, Michaelis-Menten this equation will become, because there is no alpha. So you need not to multiply the Km value with alpha. You only have alpha dash. So only the substrate concentration you need to multiply. Okay, and this alpha dash will be 1 plus I upon Ki. So I is the concentration of inhibitor, and Ki dash is the rate constant of this particular reaction in the presence of inhibitor. Now, similarly, because this is your MM equation, you can also modify your double reciprocal equation or the LB equation. So here also you only, everything will remain the same, only instead of 1, you will put some value alpha dash here because this Vmax will be decreasing. Okay. Now see the plot. Okay, so here, so the plot is very easy to interpret because they will give you the parallel line. So if you want to make a U here, you will easily understand this is uncompetitive inhibition. So in this case, if I try to annotate this, this is decreasing because this Vmax is moving up and this is a reciprocal value. Also, this is in the presence, in the absence of inhibitor, and this is the presence of inhibitor. So this will moving to the more negative value. Therefore, the Km is also decreasing. So both Vmax and Km will decrease. This is easy interpretation. Now, okay, so I need to remove this first. Fine. There is a table. Okay, so from here, if there is no inhibition, so normally you will have Michaelis-Menten equation called V is equal to V max into substrate concentration divided by Km plus substrate concentration. And the Lineweaver-Burke equation, that is double reciprocal equation, will be 1 upon V Km upon V max into 1 upon substrate concentration plus 1 upon V max. And there is no change in the values of Km and V max. If there is a reversible inhibition, either competitive, non-competitive, or uncompetitive, so what can happen? In the first case, in the competitive inhibition, only there is a change in Km value. So you will multiply this Km with the alpha, some value called alpha, and this is called apparent Km. So apparent Km will be having some high value than normal Km. And also here you will multiply this, otherwise everything is same. And you will see that this is increased apparent, APP for apparent Km. So this will increase. But there is no change in the Vmax. So Vmax is unchanged here. So they did not describe the Vmax. It will not change. Okay. So the second is pure non-competitive inhibition. So if it is pure, we know alpha will be equal to alpha dash, and therefore the Vmax will decrease and the Km will not change at all. So only the thing is Vmax will decrease. And this is apparent Vmax. Okay. Third is uncompetitive. In this case, because this is uncompetitive, both will decrease. Either Km and Vmax both are having apparent values, therefore they both will decrease. So both are decreasing, therefore this is uncompetitive. And this same table, but some modified terms you can also get from your book, Leininger 7th edition. Actually, it is given over page number 600. So if I go to Leininger and if I go to page number 600, I will have, oh, sorry, this is not working. Why? I do not know. Sorry, I just need to write 600. Okay, so this is the table. Here also there is apparent Vmax, apparent Km. If there is no inhibition, Vmax and Km will remain the same. In case of competitive inhibition, Vmax will remain Vmax, whereas this will apparent, so it will increase. So let me annotate this also. Okay, so this will increase. Apparent Km will increase in case of competitive. Uncompetitive, because here, so you always need to remember in each book, the sequence will not remain the same. It may happen that uncompetitive inhibition may come earlier. So therefore, you need to remember in uncompetitive, both are decreasing. So both will have some apparent value. So Vmax divided by this alpha dash and Km is divided by this alpha dash. Therefore, both will decrease. So make your mind like this, and more you revise, more you will able to remember all these, whether it is decreasing or increasing. In mixed type, because this is mixed, therefore this will have some apparent value. So Vmax will always decrease. Whereas if it is mixed, so the Km can increase or can decrease. Mostly it will increase, but in some cases, as we have seen, it may decrease also. Okay, so either can increase or can decrease. Fine. So the next topic of today's class is, okay, so before moving to before leaving this enzyme kinetics and before switching to regulatory enzymes, let me ask you if you are able to understand these Km, Vmax, MM equation, LB equation, Haney's equation, and plot. and there is Eddie-Hoffisti, and uh, one more, Cornish-Bowden plot. So all these plots and equations we have discussed in our previous classes. I request you all, please go through the previous lectures and uh, through your book Pathfinder, so that you are able to understand whatever we have discussed. Most probably this is our last class of enzymology, and then we will start some new topic after discussion with sir. Okay, so there is a problem. Okay, so here, if you have some query, you can ask. Hope there is no interruption in the internet and my voice is very clear and the screen is also very much visible. If you guys are having no queries, very good. So if you plot the substrate concentration in millimole with rate, that is millimole per ml per minute, either in the presence of A, that is with A, or without A. So actually A is the inhibitor. They have told you that A is the inhibitor. So in the presence of inhibitor, this is plus inhibitor. This is in the absence minus inhibitor. So you can see that in the absence of inhibitor, generally they are having more reaction rates. 5, here is they are at 3, 7.5, 5, 10, 7.5. So generally there is a difference of 2.5, except the last value, 7, 10.7, 8.3. No. Using double reciprocal plot of the data, determine the type of inhibition. So this type of question can be asked. So if you remember the inhibition types, so we have discussed irreversible inhibitions, we have discussed reversible inhibitions, and in irreversible inhibition, there is one kind that is called mechanism-based inhibitors or the suicide inhibitors. And in competitive, non-competitive, and uncompetitive, we have discussed in case of reversible inhibition. So first of all, you need to plot the LB plot and of these values, and if you get a characteristic curve, whatever we have discussed in previous discussion, okay, either this type, this type, or this type, either these types or this type, or generally this type. So this is for competitive, these are for non-competitive, and this parallel lines for uncompetitive inhibition. So if you get some characteristic curve, so based upon the shape of your curve, you can tell whether it is competitive, non-competitive, or uncompetitive inhibition. If you will try to plot these values, you will get this competitive inhibition. And if you remember the competitive inhibition curves, this is something like this. So if you extrapolate this x-axis, so because the Km value will increase, so first of all, in the presence of inhibitor, oh sorry, in the absence of inhibitor, you will get something like this. When you add some inhibitor, so the Km value will increase, so it will come here, and this point will remain the same, so they will intersect at the same point because Vmax is not changing, and you will get this type of, actually I plot this line, this will come little down here, so, and this is straight line. So if you get this kind of curve from this data, this will show the inhibition is actually the competitive type. And the second is does inhibitor A combine with the enzyme, with ES, or with both? From the discussion, you know, in case of competitive inhibition, the inhibitor will always bind to the free enzyme, whereas in mixed, it can either bind with the free enzyme or enzyme substrate complex. In uncompetitive inhibition, it will always bind with the enzyme substrate complex. So this all we have discussed. So here, because this is competitive inhibition, you are getting this shape. This will only bind with the free enzyme. So inhibitor combined by only with the free enzyme. Competitive inhibitor cannot combine with the enzyme substrate complex because the inhibitor and the substrate compete for the same binding site. And they have also given the reason because they are having only one site over the enzyme, therefore they can bind only with the free enzyme. Okay. So that's all this type of problems you can face. Now our next topic is regulatory enzymes or allosteric enzymes. Okay, so what are regulatory enzymes? So there are various pathways. So we have discussed so many different types of enzymes. Okay, before starting this topic, actually I left with one more thing. So I need to tell you. Okay, so actually we did not discuss few examples. So for example, we have discussed irreversible inhibition, and in irreversible inhibition we have discussed suicide inhibition, and suicide inhibition is given in your book also. So if you try to find it. Okay, so not here. Okay, so here is a special class of reversible inhibitor is actually called suicide inactivators or mechanism based inactivators. We had discussed it yesterday when I told you that what will happen actually. So the inhibitor will bind to the enzyme just like the substrate will bind. So enzyme think this is the normal substrate, but it was actually the inhibitor with which the enzyme has bind. Now what will happen? The enzyme will not be inhibited immediately by the inhibitor. After binding, the enzyme will process the inhibitor in the same way as it will process the substrate into the to make the product. So, but in this processing, enzyme is unaware of the fact that the inhibitor will be converted into more reactive intermediate and it will permanently block the enzyme. Therefore, this is called suicide inhibition or the mechanism based inhibition. Okay, so one example is given here, which is very common and used to treat some kind of so. Okay, so chymotrypsin can be inhibited with the help of DIFP, diisopropyl fluorophosphate. This we have discussed yesterday. DIPF, sarin, that is also called nerve gas, and other inhibitors like parathion. They all are irreversible inhibitors. But one very interesting example, which is used to cure the African sleeping sickness. Okay, so this is a kind of mechanism based inhibitor, or you call it suicide inhibitor. Okay, so this African trypanosomiasis is a disease which is actually caused by some protist, single cell eukaryotes that are called trypanosomes. So something you know about this disease, you can also get information from this page number actually 603. It is given in 7th edition of Leninzer. So I want to tell you how it is inhibiting the enzyme actually, because this is having some mechanism based enzyme inactivator or suicide inactivator. So a vulnerable point in trypanosome metabolism is the pathway of polyamine biosynthesis. So how you will kill these trypanosomes or the single cell eukaryotes with the help of this drug. So because these are having, they need to synthesize some polyamines. And in polyamine biosynthesis, they are using this spermin and spermidine, which is involved in DNA packaging, just like histones in higher eukaryotes. So these polyamines, for example, spermin and spermidine, which is used in single cell organisms, or in trypanosomes, which are the causative agent of African sleeping sickness or trypanosomiasis. So in some competitive examination, you can get a direct question. So the trypanosomiasis is a disease due to which pathogen? So it is generally due to single cell eukaryotes, and the disease is called trypanosomiasis. Okay, so involved in DNA packaging, these spermin and spermidine, and are required in large amounts in the rapidly dividing cells. Suppose the eukaryotic cells need to divide very often. So because these are single cell eukaryotes, so they will divide very fast. Therefore, they need to make the DNA and need to pack the DNA. Therefore, they need to biosynthesize these polyamines. Therefore, you need to target some pathway of their polyamine biosynthesis. So first step in their synthesis is catalyzed by ornithine decarboxylase enzyme. This ornithine decarboxylase you also have discussed when we are in our previous classes. Okay, is an enzyme that required for its function as an coenzyme called pyridoxal phosphate. So pyridoxal phosphate PLP is generally used in amino group transfers. Pyridoxal phosphate derived from the vitamin B6, this also we have discussed, form a covalent bond with the amino acid substrate of the reaction. It is involved in the electron sink and facilitate variety of reactions. In mammalian cell, this ornithine decarboxylase undergo rapid turnover. So because this enzyme is having very high turnover, you know turnover? So the turnover is synthesized very, means very often because it is required in high amount. Is a rapid constant round of enzyme degradation and synthesis. So it will be degraded fast, it will be synthesized again and so on. And some trypanosomes also, however, this enzyme is stable. Okay, so this is the main difference between us and the eukaryotic single-celled eukaryotes, which are the causative agents of African sleeping sickness. Because in the single-celled eukaryotes or the trypanosomes, this enzyme is not very often synthesis. It is very stable. So once it is formed, it will remain throughout the through for most of the time. But because in eukaryotes, this will have a rapid turnover. So suppose we take an inhibitor as a drug, it will inhibit some enzyme, but there is nothing to worry about because we are having a rapid turnover. We will make new enzymes. Okay, but it is harmful to the trypanosome because it will not having that much rapid turnover of this particular enzyme, ornithine decarboxylase. So first of all, you need to remember the name of the enzyme. Ornithine decarboxylase is the targeted enzyme, and this will undergo mechanism-based inhibitor or inhibition or suicide inhibition. So, so therefore, it will adversely affect the parasite. So these are parasites shown here. These are trypanosomes. Here is the reaction, how it is. So pyridoxal phosphate, with the help of pyridoxal phosphate, which is derived from vitamin B6, it will convert ornithine into this form, some CYP base form after dehydration and some decarboxylation. Okay. And then again, when water molecule is added, so this will be released in the form of putrescine. So this decarboxylation step or reaction is with the help of this enzyme ornithine decarboxylase. Now, what is the drug which is generally used to treat the African sleeping sickness is actually the suicide inactivator, which is designed in such a way. So this is difluoromethyl ornithine. Okay, that is called DFMO. So this DFMO is used as a drug to permanently inhibit or irreversibly inhibit this ornithine decarboxylase enzyme. DFMO is relatively inert in the solution, so therefore it will not, it will stable, it will be stable. It will not change. When it binds to the ornithine decarboxylase, how the enzyme is quickly inactivated. The inhibitor acts by providing an alternative electron sink in the form of two strategically placed fluorine atoms. Because it is having two fluorine atoms, therefore it will accept the electrons and will act as an electron sink. Okay, so the rest you can read from the book. The S of the cysteine residue, because the cysteine is having SH group, so this thiol group or the sulfur at the enzyme active site then forms a covalent bond with the. This is also very important how this is happening. So this DFMO is irreversibly inactivating ornithine decarboxylase, first thing. So therefore, this can be used to treat the African sleeping sickness because in trypanosome, the decarboxylase enzyme is very stable and do not have a very rapid turnover as in case of higher eukaryotes. And what is the mechanism of action? So it will make a covalent complex or covalent bond with the cysteine amino acid. And the cysteine is having some side group sulfur. So the sulfur will bind with this fluorine of this DFMO or difluoromethyl ornithine. So the result of displacement of fluorine atom, so sulfur or the cysteine residue in the enzyme active site will form a covalent complex with the highly reactive PLP inhibitor adduct, and then essentially a reversible reaction. So this is an irreversible inhibition, and therefore enzyme on reaction mechanism to kill the trypanosome. Okay. One more thing you need to remember, this is also a very important question. So African sleeping sickness is actually caused by Trypanosoma brucei gambiense. Okay. There is one more Trypanosoma cruzi, I think, which is responsible for some other disease you can find from the internet. Okay. But you need to remember this Trypanosoma brucei gambiense is the actual agent, single cell eukaryotic trypanosome for African sleeping sickness. This can be your match the column type of question. Now we will move to page number, so we are at 606. Now we will move to 625. So 625. Also, there is one more thing I need to tell you in this enzyme logic class. So when some people want to establish the mechanism of action of this enzyme, the enzyme name is given somewhere. Okay, so hexokinase we have discussed in our very first class of enzymology. And to prove that this hexokinase is having some induced fit form, so there are two models we have discussed in our classes. One was lock and key hypothesis model, and one is induced fit. To prove the induced fit, the enzyme hexokinase was used, and this hexokinase can bind to the xylose and glucose. Generally, it binds with glucose, but when it can bind with xylose also, because xylose is a 5-carbon sugar, and this is a 6-carbon sugar. So what will happen? Let's read these few lines so that you are able to understand. This model been reinforced by kinetic studies, 5-carbon sugar that is called xylose. So this is a direct question. Can we ask somewhere? So xylose is a 5-carbon sugar which is used to, okay, stereochemically similar to the glucose. If you try to look at the structure, it is aldehyde group at very first carbon, COH. So OH is on the same side, then this OH on the left hand side, this OH on the right hand side. Then last is CS2OH. Only this will not have this 5th carbon, therefore 1 carbon short. Okay, otherwise it is very stereochemically similar to the glucose, but 1 carbon shorter. Bind to the hexokinase enzyme, but in a position where it cannot be phosphorylated. So how the scientist is able to know that the hexokinase will only bind to the hexose sugars, and this is a kind of induced fit binding. So after binding, it will the enzyme is undergoing some conformational change to fit more efficiently with the substrate glucose. Nevertheless, addition of xylose to the reaction mixture increases the rate of ATP hydrolysis. Okay, so see, what is the function of hexokinase? Hexokinase is a kind of phosphate transferase which will transfer the phosphate group, gamma phosphate group, from the ATP over this glucose residue or any hexose sugar, because this is hexokinase, so any hexose sugar can be phosphorylated. But if you bind some 5-carbon sugar, which is very stereochemically similar to this glucose or 6-carbon sugar, it will not undergo the reaction. It cannot be phosphorylated, but What is seen in the solution, the rate of ATP hydrolysis will increase. Okay. So the ATP will be hydrolyzed, but the phosphate will not be transferred over this xylose. Evidently, the binding of xylose is sufficient to induce a change in hexokinase to activate the conformation. So it can be proved that the enzyme is activated. So obviously, the binding of xylose is sufficient to induce a conformational change in the hexokinase so that it can hydrolyze this ATP molecules. Okay, and the enzyme is therefore tricked into a phosphorylating water molecule. Okay, so you can think like the water molecules will be phosphorylated. The hexokinase reaction also illustrate that enzyme specificity is not always a simple matter of binding one compound but not other. And this will also prove that the enzyme will specifically bond to the substrate. But if the some other molecule is stereochemically similar, so it can also bind with that particular molecule also. And therefore, there come the concept of inhibitors, because some inhibitors are very similar to the substrate. Therefore, they can bind with the same site, same active site where the substrate will bind. For example, we have seen the case of competitive inhibitors. So the hexokinase reaction also illustrates that the enzyme specificity is not always a simple matter of binding. So the enzyme can bind with the very similar molecules also, but with not the another one. So it will not undergoing the same. It will not lead to the formation of phosphorylated product. Xylose. So in the case of hexokinase, specificity is observed not in the formation of enzyme substrate complex, but in the relative rate of subsequent catalytic reaction of the steps. Reaction rate increases greatly in the presence of substrate, for example, glucose that is able to accept the phosphorus. And it has been seen that the reaction rate will further increase in the presence of glucose because it will able to accept the phosphate residues from the ATP, that is the gamma phosphate actually, but it will not able to accept the phosphate residue, but it will able to activate the enzyme. Therefore, hexokinase use several catalytic strategies, just like chymotrypsin. Okay, so you can read the text further. Only thing I want to tell you from this discussion or from this theory, actually this xylose is being used to prove that this hexokinase is undergoing some induced fit, so that it is able to activate the hexokinase. It will means hydrolyze the ATP molecules, but it will not able to phosphorylate the xylose because the enzymes are very specific for their substrates. Okay, so rest is. Then we will move to another page 641, which is actually. Okay, so here you can see that there are catalytic subunit, there is regulatory subunit. So these type of enzymes are called regulatory enzymes or allosteric enzyme because they are having two different sites for binding of modulators. Okay, so the modulator can bind, so this catalytic site is generally binding with the substrate. Okay, but this modulator is having some kind of effect over the enzyme. To my surprise, there is no query in today's class. Okay, so let me quickly finish the topic. Actually, this is regulatory enzymes, which is very easy to understand with the help of this cartoon. So you can see that there is a box type conformation of this catalytic site, but this is a substrate is having some different shape. So, and also this is having some different shape, regulatory subunit. So mostly the regulatory enzymes or allosteric enzymes, allo means different sites. So both the substrate and modulator can be positive modulator or a negative modulator. Okay. So what is the role of positive and negative modulator? If the negative modulator will bind to the this regulatory subunit, it will it will destroy this binding site of this catalytic subunit, so that substrate is not able to bind. Whereas if it is a positive modulator, it will enhance it will undergoing, this enzyme will undergoing some conformational change, so that is this site, the active site, is modified into a form which can more efficiently bind with the substrate molecule. So it is very easy to interpret that these regulatory subunits are having some role in the allosteric enzymes, so that they either increase the rate of reaction or decrease the rate of reaction. Okay, and both are having different sites, therefore called allosteric enzymes. And most important point to remember that they do not follow the equation, kinetics. Okay, so that they can bind like this. So this will make an active enzyme substrate complex. Because this is the complete enzyme and this is the substrate, they are bound, they are able to bind, they can make the active complex. The kinetic properties of allosteric enzyme diverge from the Michaelis-Menten behavior. How? We will discuss with the help of some plots. So see here, this black line is showing something which is happening in case of normal allosteric enzymes. So this is S-shaped curve. Whereas in case of the enzymes which are following the Michaelis-Menten equation, they are showing some hyperbolic type of plot. Not this is very hyperbolic, but they are actually showing this kind of way. So if they are showing something like this is called hyperbola. Okay, but here this is not shown. They will diverge from this equation and plot. Therefore, they will generally have this S-shaped curve. If you increase the other high activity of R state, this is resting state, and this resting state is, if it is having high activity, so the normal line, which is the S-shaped curve, will be shifted into this form. Otherwise, if there is low activity, this will be shifted to the lower side. Okay, so this can shift like this. Also, if there is a change or the modulation in the enzyme, in the presence of some positive modulator and negative modulator, so they can vary. So either they will move like this, and ultimately they will have this same line, which is actually in the R state. And low activity, this is the tense state. So in the presence of negative modulator also, they will also show this kind of if you try to find the half Vmax and Km values for these all enhanced regulatory enzymes, so this you can find mostly in presence of negative modulator, they will have high values of these Km, or what you call K half. Okay. So let us discuss the Pathfinder portion so that we can finish our. So the regulatory enzyme exhibit increased or decreased catalytic activity in response to specific signal, because these are regulatory enzymes and uh, they are under some kind of signal. So based upon signal, they can modulate the enzyme activity, or they can uh, deactivate the enzyme or decrease the enzyme activity. Catalytic activities of regulatory enzyme can be modulated through the reversible non-covalent binding. Okay, so this you need to underline. This is reversible. So this is reversible non-covalent binding by the regulatory compound or by the reversible covalent modification. So two type of modulation can happen in the regulatory enzyme. Either one is reversible non-covalent binding or the reversible covalent modification. Okay, so either covalent or non-covalent, both can happen, but overall there is a change in the enzyme activity. Sorry, I need to remove this first. So regulation by the non-covalent modification first. So first of all, we will discuss this non-covalent modification, and then we will discuss the covalent modification, which is reversible actually. So the allosteric enzyme from the Greek word allo for different place because they are having different sites for binding of enzyme, substrate and inhibitor. Not inhibitors, these are regulators actually. So here they are called modulators, either positive modulators or negative modulators. So they are having different sites or class of regulatory enzyme which increase or decrease the catalytic activity in response to the certain signals. And actually they are under some signal. Uh, oh, every time I need to remove the drawing actually. This is bad. Okay. So these are called modulators or effectors. They can be positive or negative modulators and effectors. Modulators for the allosteric enzymes may be inhibitory or stimulatory. Inhibitory, they are decreasing. Stimulatory, they are increasing the rate of reaction. Often the modulator is the substrate itself. In some cases, you will find that the substrate is the modulator itself, and regulatory enzymes for which the substrate and the modulator are identical, they're called homotropic. Whereas if the modulator is different, then the enzyme is said to be heterotropic. So this is understandable. Then this we have discussed. Now we will move to okay, here something missed. No, not at all. So here we are at page number 124. Most allosteric enzymes are oligomeric. So oligomeric in the sense they are made up of multiple subunits. Mostly, at least they have two subunits. One is catalytic subunit, one is regulatory subunit, or in some cases they may have more than two subunits. So oligomeric. Allosteric enzymes of heterotropic nature means what are heterotropic? If the modulator is not the substrate but some other molecule. Heterotropic nature, they have two functionally different binding sites. Obviously, they will have two different binding sites where the modulator will bind, and other is the active site where substrate will bind. One site, a catalytic site, where the substrate bind and catalyzes the reaction. Other site is where the allosteric or the regulatory site, which is molecule bind, that is called modulator. Modulator molecules are of two type, positive and activators, and enhances the enzyme activity, and there is negative modulators also which are called inhibitors actually. So whatever we have discussed in inhibition, they are negative modulators of those enzymes which inhibit the enzyme activity. Binding of the modulator causes conformational changes in the allosteric enzyme that influence their catalytic activity. So what will happen, the substrate is not able to efficiently bind to the enzyme because they will destroy the shape of active site. Enzymes with the several modulators generally have different specific binding sites for each. For example, there are oligomeric enzymes. Oligomeric enzymes, when they are made up of multiple subunits. So some enzyme complexes are reported where there are more than two enzyme subunits, and they will make an complex. One one common example is fatty acid synthase, for example. Okay, so this fatty acid synthase is made up of 7, 8 subunits. So all the subunits may be modulated with different modulators. So in these cases, it can happen that more than two sites over the enzyme can be present. Binding of modulator causes a conformational change in the allosteric enzyme and activity. Okay. Now the allosteric enzyme do not obey Michaelis-Menten kinetics. So this you need to underline, they do not obey, they do not follow the equation, and therefore the plot cannot be derived from the data of the allosteric enzymes. Generally, they are having this S-shaped kinetics. They do exhibit saturation with the substrate when S substrate is sufficiently high, produces sigmoid saturations, the sigma or the S-shaped curve, rather than a hyperbolic curve typical of non-regulatory enzymes. Sigmoid kinetic behavior generally reflect cooperative interaction. You know, cooperative is something, for example, in case of hemoglobin, when hemoglobin bind with one oxygen molecule, and when bound to another second oxygen molecule, third oxygen, one hemoglobin molecule or the protein can bind with 4 oxygen molecules, but each time it is binding with the oxygen molecule with a high affinity. So one oxygen molecule will bind to the hemoglobin with very difficulty, but when the second molecule and third molecule and the lastly molecule will bind, they will not face any difficulty, because the affinity of the enzyme or the hemoglobin or the protein for the substrate, that is oxygen actually, will increase. This is called cooperative binding. So because the sigmoid curve is forming there also, in case of hemoglobin saturation curve, you will see this sigmoid curve. So this is actually a kind of saturation kinetics. Okay, so it will show reflect some cooperative interaction or behavior between the protein subunits. The principles are particularly well illustrated by the non-enzyme when the oxygen binding to the hemoglobin. This is given here, so this is an example. Sigmoid kinetic behavior is explained by the concerted and sequential models of subunit interaction. The sequential and concerted model, this I think this you have been taught by Nitin sir in your protein class. Okay. So there are two type of models. One is concerted model, one is sequential model. Concerted sequential model is when the oxygen molecule will bind to the each subunit sequentially in a fixed. Okay, wait 1 minute. I'm telling you the page number where it is given actually. So if some of you, I know some students are new actually, including Anchal. So it is actually given over page number. So hemoglobin is, if you are having 7th edition of Pathfinder, so page number 37, they have discussed about hemoglobin, and over page number 39, they have made this sigmoid curve, and this myoglobin is actually having this hyperbolic curve, whereas hemoglobin is having a sigmoid curve. This is the major difference between hemoglobin and myoglobin. Myoglobin do not show the cooperative behavior, whereas hemoglobin is showing the cooperative behavior. And at the next page, that is page number 41 actually, they have given the shift in the presence of bisphosphoglycerate. Okay, so Bohr effect and all these things has been discussed. And there are some behavior of allosteric enzymes or protein given over page number 42 and 43. So there are two models. One is sequential model, one is concerted model. So in the concerted model, each enzyme is having some shape. So it will be reverted into all forms, whereas sequential model is something when the oxygen molecule will bind with one hemoglobin, it will undergoing some conformational change and the rest will remain as such. Okay, in a sequential fashion. So page number 42 and 43 is describing these concerted and sequential models of allosteric enzymes. Then this is how you can calculate this K half from this half Vmax value in case of sigmoid curve also. And generally they will show positive and negative. Generally positive influences when this curve is shifting towards the left hand side, and negative is when it is shifting towards the right hand side or lower side. Regulation by reversible covalent modification. So this is the second type. So because this is reversible covalent bond modification, so some covalent bond will form, but they are all reversible. So that's how these allosteric enzymes can be regulated. So activity of some of the regulatory enzymes is modulated with the reversible covalent modification, as we will discuss some of the examples. Covalent modifications include phosphorylation. Okay, so covalent is something, for example, over serine, threonine, and tyrosine, phosphorylation, adenylation, methylation, uridylation, and ADP ribosylation. Phosphorylation is a common type of covalent modification. So phosphorylation is the basic type of modification. We have discussed many times phosphorylation, dephosphorylation, and cell signaling also. So this is the basic type, and most often it is happening in the case of allosteric enzymes. So in some cases, the phosphorylated enzymes will be activated. In some cases, phosphorylated enzymes will be deactivated. So not always like that. The phosphorylation will lead to the activation. This will not happen always. Okay. So phosphorylation is accomplished by the protein kinase. So there is kinase, there is phosphatase. So they will regulate the kinase. Kinase will phosphorylate. Phosphatase will remove the phosphate group from the particular residue. An important example of regulation is given by phosphorylation occurring in glycogen phosphorylase enzyme, which is present in muscles and liver. What is the function of glycogen phosphorylase? So glycogen phosphorylase is an enzyme we also discussed in the cell signaling class. So glycogen phosphorylase and glycogen synthase will function in opposite way. Glycogen phosphorylase will be activated after phosphorylation, whereas glycogen synthase will be inactivated. So glycogen phosphorylase will catalyze the glycogenolysis. What is glycogenolysis? Breakdown of glycogen. Whereas glycogen synthase will lead to the glycogenesis, means glycogen will be synthesized. Glycogen phosphorylase occurs in two forms. One is A form and one is B form. So A is active form, B is less active form, not the inactive form actually. In glycogen phosphorylase A, or the active form, there is a specific serine residue. If you phosphorylate the serine residue, it will be A form. If you remove this phosphate group from the serine residue, it will be B form. So converted to less active glycogen phosphorylase B form after enzymatic loss of these phosphoryl group from the serine residue. Okay, glycogen phosphorylase B can be reconverted into the glycogen phosphorylase A by the action of kinase, and can be converted into A form. Can be converted into A form with the help of kinase, and will be converted back into the B form with the help of some phosphatase, not phosphorylase. So phosphorylase and phosphatase both are different. Phosphorylase are actually kinases. Phosphatases are the enzyme which will remove the phosphate group. Some examples are given. Glycogen phosphorylase modification is phosphorylation, and it will be activated after phosphorylation. Glycogen synthase, phosphorylation can happen, but it will be inactivated after this phosphorylation. Acetyl coenzyme carboxylase can undergo phosphorylation also. Glutamine synthase is present in E. coli, and it is undergoing some adenylation. And nitrogenase is undergoing some ADP ribosylation. So these type of modifications are found in these enzymes, which will either activate or inactivate these particular enzymes. Uh, Now, how these regulatory enzymes are regulated. So there is some kind of signaling. So this is called feedback signal. So if uh, suppose there is a regulatory pathway, the metabolic pathway, where the substrate A is getting converted into intermediate B and C, and finally into a product D. If this product D is accumulated, so this product D is accumulated or present in excess, will go back and will inhibit the first enzyme of this particular metabolic pathway, so that more enzyme will not be synthesized, and thus there is no need, because there is sufficient product D present. Therefore, the first enzyme itself will be inhibited. This is called negative feedback. This is also called end product inhibition, because the last product is inhibiting the first enzyme. End product is a biosynthetic pathway, inhibit the activity of the first enzyme itself, which is regulatory enzyme most often. For example, in case of glycolysis, the hexokinase is a kind of, or the glucose 6-phosphate is a kind of enzyme which is regulatory in nature. That is unique to that particular pathway, thus controlling the production of the end product. For example, D here, the first enzyme in the pathway is an allosteric enzyme, or the regulatory enzyme. So the allosteric site will bind to the end product D, and therefore it will work like an effector, therefore will alter its active site so that it cannot mediate the enzymatic reaction further. It is a negative regulation, so therefore it is called negative feedback or end product inhibition. It prevents, because here this product is acting like an inhibitor. It prevents the enzyme from acting. Enzyme can be subjected to positive regulation also. If this is consumed, then it will be relieved, and therefore this will undergo some positive regulation also, in which enzyme activity is stimulated by the regulatory molecules, rather than being suppressed. So some positive regulators, some positive molecules or a modulator will come over this regulatory enzyme or allosteric enzyme 1, and they will modulate the pathway if this excess D is consumed in the body or within the cell. Then there is another type that is called feedback loops, in which the first enzyme is not inhibited. Instead, some other enzyme of the pathway is allosteric in nature, and that is affected. So, in a metabolic pathway, because there are long branches leading to the synthesis of two products. So, suppose there is a product called A1, there is a product called A2. Different products, each end product can control its own synthesis without affecting the other. So in this way, if it will inhibit the, suppose this product will inhibit the enzyme A, so it will also affect the synthesis of product A2. Therefore, they will have some different feedback loop kind of regulation, so that they can control their own synthesis or accumulation without affecting the other products. So therefore, the feedback inhibition is different from feedback repression. Amount of enzymes synthesized by the cell are determined by the transcription regulation and inhibitory feedback system, in which the end product produced in the metabolic pathway acts as a co-repressor, and it will work with the repressor molecule, and the repressor, the synthesis of the enzyme that is required at the earliest stages of the pathway called feedback repressor. Instead of feedback inhibition, there is another form also, feedback repression. So this feedback repression, you can, if you read it from here, or you can find it in more details from the internet. Okay, then there is isoenzymes and alloenzymes, riboenzymes. These all DNA enzymes. All these we have discussed in our previous classes. So we finished this enzyme portion from your Pathfinder. Okay.